Hello students, this is module 4.1.3, functions of DNA. DNA has two main functions. DNA, primarily it codes for proteins, and this accomplishes both of the functions. The first function is gene expression, and the way that a gene is expressed is by making a protein. And gene expression allows cells to carry on their day-to-day functions by making the proteins that are needed for the occurrences and normal functioning of the cell. The second function that DNA accomplishes is heredity. This is a long-term function. This is, of course, as you learned, occurs in cell division, and so all reproduction is based on cell division, whether it be sexual or asexual. So those are the two main functions that you should think of: making proteins for day-to-day use, and also heredity. And DNA accomplishes both of these. Let's learn some vocabulary, some basic terminology. Understand that a gene is a segment of DNA that codes for a protein. So one chromosome is one DNA molecule, and one little section of that DNA molecule that codes for a single product is called a gene. Now, genes, of course, are elements of focus here. So genes are able to replicate. They can copy themselves. Genes can mutate. They don't copy themselves perfectly every time. If they did, we wouldn't really have any evolution. Genes store information, and genes use that information to direct synthesis of structural and regulatory proteins. And in some respects, DNA is like a reference library. I remember when I was a little boy, I loved to go to the reference section of the library because I was interested in all sorts of interesting creatures, snakes and butterflies and things like that. And they had wonderful reference books. And I always wanted to check those books out, but the librarian would say, no, if you can't check it out. As a college student, you know that if you need information from the reference section of the library, that what you have to do is copy it and take it out. And in that respect, DNA is like a reference library. So DNA stays in the nucleus, and the information, if it's needed in the cytoplasm, is copied and taken out. And of course, in a library, you have a strict librarian to enforce that. But of course, that's not how it works in the DNA. Typically, though, the way it does work in DNA is that one gene's worth of DNA will be copied into an intermediary molecule called RNA, and the RNA transcript, or the RNA copy, will be transported out to the cytoplasm, where it will be used to make as many copies as are needed of a particular protein. So now we've introduced the role of this new molecule. Let's learn a little bit about it. RNA stands for ribonucleic acid. It is very similar to the structure of DNA with a few exceptions. RNA is often single-stranded. RNA has the base uracil replace thymine, so there's no T in RNA. Instead, there's a U. RNA has a different sugar in the backbone. The sugar of RNA is ribose, and of course of DNA, the sugar was deoxyribose. And you might guess, or you can refer back to the slide where I showed you the exact structure of these two sugars. But the difference is, of course, that deoxyribose has one less oxygen. And RNA is shorter in length. DNA is as long as a chromosome. RNA is usually as long as a gene at its longest. The major function of RNA is its role in protein synthesis. But there are, in fact, multiple types of RNA, and they have varied functions. We're going to learn three main types. But before we do, let's make sure you really understand RNA. And a good way to clarify understanding is to compare it to something that you know. You know a little bit about DNA, so we can use this format of comparative table, and we can compare a number of features. The first feature we'll compare are the bases that are present. we can see in DNA there's A and T and C and G. Of course, in RNA, there is no T. T is replaced by U, uracil, and so it's A and U and C and G. The 5-carbon sugar in DNA is deoxyribose. The 5-carbon sugar in RNA is ribose. The number of strands, DNA is typically double-stranded, while RNA is typically single-stranded. In other words, DNA has 2 backbones and looks like a ladder. RNA looks like half a ladder, has 1 backbone. How long are the respective molecules? Well, DNA is very long. It's as long as 1 chromosome. RNA is short. The longest RNA is as long as 1 gene, which is a fraction of the length of 1 chromosome. The location, of course, DNA is like a reference library, so it stays in the nucleus. And the reason for that, by the way, I don't think I mentioned before, is because the cytoplasm is a very harsh chemical environment and DNA could be degraded. And remember, DNA has a second function besides making proteins, and that's inheritance. And if the DNA is degraded and changes too rapidly, that would lead to chaos. And so that's why it stays there in the nucleus like a reference library. Of course, RNA is made in the nucleus by copying DNA, and then it's exported to the cytoplasm where it's used to make a protein. Here's a pictorial comparison. On the top is DNA, 2 strands, RNA on the bottom, 1 strand. We can also see, if we look very carefully at the bases, that there's U's down here, no U's up here in DNA. Instead, thymine replaces uracil, which is the U. So that just gives us some idea of the comparison. I am going to quickly, in an overview fashion, talk about the 3 major kinds of RNA. The first type of RNA is called mRNA. It stands for messenger RNA. And this is the kind of RNA that I'm talking about that copies 1 gene's worth of DNA in the nucleus and takes that message out to the cytoplasm. And so that's its function. The origin of mRNA is it's copied from one of the strands of DNA in the nucleus. If you don't know what the word template means, I say it's copied from a DNA template. Template is something you copy. And the second type of RNA is ribosomal RNA, or rRNA. This RNA is a structural molecule. It serves as a structural component of ribosomes. So ribosomes are made of ribosomal RNA and proteins. And so you will recall that ribosomes are the structures in the cytoplasm of cells that serve as the sites of protein synthesis. There are actually multiple subunits, 2 subunits in each ribosome. They're made of a large component and a small component called the large and small subunits. And just reminding you, I just said it, but ribosomes are the site of protein synthesis. And that becomes very important because you will recall that one of the major functions of DNA is to guide the synthesis of proteins. The third and last type of RNA that we're going to consider is called transfer RNA, or tRNA. Now, this has a complex function that we'll look at in more detail later, but for right now, let's just say that tRNA is a molecule that translates from the language of nucleotides to the language of amino acids. It goes from DNA to protein, from nucleic acids to protein. And as I say here, we'll talk more about that later. Right now, I want to give you a diagram that's very important. This is an overview of some of the main processes of DNA, and you should take this to heart and learn about this diagram. So let me get my pen, and this diagram is called the central dogma of DNA. The central dogma is sometimes called the central dogma of molecular biology or of cell biology. And what this does is it details the flow of information, and I'll just abbreviate information, in the cell. And it consists of, and you need to remember this isn't actually part of it, so this is just to the side over here in parentheses. It has 3 molecules and 3 processes. And let me draw this for you. And you should be able to draw and explain it in a similar fashion. So the three molecules are DNA. I'm going to make a long arrow. RNA. I'm going to make a long arrow. And protein. So these are the three molecules. And the arrows actually represent the processes. So the three arrows I've made. The first process is called replication, DNA replication. This is one you already know about. This happens in S phase of interphase when chromosomes are duplicated. A second process, rather not a function, is transcription. And a third is translation. So that's the bare bones. That's the three molecules and the three processes. Of course, this diagram is going to serve as an overview for our next lecture topics. We're going to talk about the processes, replication, transcription, and translation. But right here, I want to mention several more things. One thing I want to mention is that there is another process which happens in some cells, and I'm going to draw this as a dotted arrow, and it goes from RNA to DNA, and this is called reverse transcription. This is done only by retroviruses, or it's done commonly by retroviruses, I should say. Retro means going backwards, and so going from RNA to DNA is considered a backwards flow of information of sorts. An example of a retrovirus would include HIV, which of course causes AIDS. And so if we looked at the HIV virus, which stands for human immunodeficiency virus, and it causes the AIDS, acquired immunodeficiency syndrome. If we look at that virus, it has as its genetic material RNA, and that RNA is injected into cells, and then that RNA has to go through the process of reverse transcription, which requires a special enzyme, reverse transcriptase, which can make DNA, which then can take over the cell. But the really critical thing that I want you to know with the central dogma are some of the functions here. And so with the three processes, the two major functions of DNA are associated. You've already learned these functions. I'm going to put here with replication, heredity. We know that DNA is replicated in S phase of interphase in preparation for cell division, and that reproduction is based on cell division. The second major function is associated with the processes of transcription and translation. And this is, of course, protein synthesis. Protein synthesis is also like gene expression. The way that a gene is expressed is by making a protein. So that's an alternate name for protein synthesis, is gene expression. So now we have three molecules, three processes, and now we have two functions that are found in the central dogma as well. And so this really does have a great deal of information. This is hard-won information that molecular biologists have had to work very hard to get, and we just present it to you here in Biology 101, just another service that we offer. Here is a alternative expression. I like the drawing that I made better than this, but certainly it kind of gives you the same outline. This information here, the central dogma, outlines the flow of information in the cell, consists of three molecules and three processes represented by arrows. You should be able to draw and explain it. That is a minimal expectation that I have of you. It's a very important part of this lecture, and so you should know the central dogma. The processes of the central dogma will provide the outline for our next three lecture topics: replication, transcription, and translation. I already mentioned about reverse transcriptase, reverse transcription and reverse transcriptase, the enzyme that carries that out. I'm not going to go over it again. We know that the central dogma is not perfect. We know that it's incomplete, but it gives us a conceptual model to begin understanding DNA. So again, the processes of the central dogma. This is just a brief definition. You should be able to understand and explain each of these processes, not only from this brief explanation I'm about to give you, but also from the more in-depth explanations, which will come later. Replication is defined as making a copy of DNA, and this occurs in S phase of interphase. So we make chromosomes replicated. Let me just remind you here that we can have an unreplicated chromosome, which consists of one chromatid and one centromere, and then we have replication, and this will make the chromosome replicated. So we have twice as much DNA. We have two chromatids, sister chromatids, one being the copy of the other. And so that's kind of where it fits into what you already know. Second major function, of course, is transcription. Transcription is defined as copying one gene's worth of DNA into RNA to use the information later in the cytoplasm to make a protein. So that gene is expressed by making the protein. And that's transcription. If you know what a transcript is, it's a copy. A college transcript is a copy of your work here at the college. And of course, if you request a transcript, the people in the administration office, the registrar's office, will copy your transcript and send it to the institution. And so that's what's happening here too. A copy of one gene's worth of DNA is being made in transcription. And translation, we're going from one type of molecule, from nucleic acids, to another, to proteins. And so it's making a sequence of amino acids, that is a protein, from the sequence of nucleotides in the RNA transcript. And we also, in the central dogma, in addition to talking about those three processes, told you about the two functions, and so there they are. Heredity is accomplished by replication. Protein synthesis, gene expression, is accomplished by transcription and translation. And you should be able to include these functions in your schematic drawing, as I did earlier. And here, finally, just to wrap up this module, we'll show you a pictorial version of the central dogma. And there's something kind of interesting here I want you to consider, that DNA is the genotype and that protein is the phenotype. So we are an expression of proteins. When you look in the mirror, you don't see DNA, you see your proteins. And this is actually showing you the processes by which we go from DNA to protein. That's transcription and translation. And with that, we'll end this module.